We have synthesized fats, and now we're going to talk about the synthesis of the nitrogen compounds, particularly the amino acids, and then a couple other important nitrogen compounds, including the nucleotides that are found. When we look at the synthesis of nitrogen compounds, the first thing that we really need to get is nitrogen itself. Atmospheric nitrogen is the largest source of nitrogen, although atmospheric nitrogen is very unreactive. If you think about the not the three-dimensional structure, but the electron structure of a nitrogen, it's a triple bond. That is hugely energetic to break. And so you've got really two ways to break down nitrogen in the natural process. One is lightning. If you are in Georgia watching a golfing game, the golfing tournament this weekend, you are a little bit too close to the lightning. Did you see that on the news? Man, I saw it hit that tree and it started raining, so everybody went under a tree. Never go under a tree during a thunderstorm. It was a bad move. But the electricity involved in a lightning strike can actually react with the nitrogen to create the nitrates and nitrites, and it can come down as nitric acid. Now, that's not how most nitrogen is fixed into the biological system, although that is one method. We can't do it, okay? Plants can't do it, animals can't do it, but little tiny organisms on the roots of plants can actually react with the nitrogen and what's called fix it or break the triple bond to create ammonia that can be incorporated into synthetic pathways. We can do this industrially with nitrogen and hydrogen under what's called the Haber process. It's really intense. You have to use a lot of pressure to get that done. And a lot of our fertilizers that we use this process to create nitrogen. And so we will enhance soil with this organic nitrogen from that particular source. But the majority is done in the microorganisms in the roots. We're going to use an enzyme called a nitrogenase. To break it with enzymes, to take nitrogen and produce ammonia, we would need 6 electrons and 6 protons to make ammonia, although there's a side reaction that also makes hydrogen. So the overall process is 8 electrons and 16 ATPs. So if you think about the nitrogen in that triple bond, 16 ATPs is a lot of energy, plus the energy of the electrons. If you think about that, 2 electrons, when we carried it as NADH, made a whole bunch of ATPs. And so this is a very energetic process. The first enzyme in this is a reductase. And what we actually have is a iron sulfur center that can hold a lot of electrons. Also contains an ADP-ATP binding site. And what the electrons or the ATP does is to change the conformation to allow the electrons to be accepted and then donated into the next enzyme. The electrons actually come from ferredoxin. Where was ferredoxin found? Where do we get electrons in ferredoxin? It's one of the pathways we looked at. It doesn't happen in the human body. Anybody got a guess? No guess. Oh, ferredoxin had a lot of things going for it. We had to know a lot of things that ferredoxin did. Oh my goodness. It's in photosynthesis. Is it in photosynthesis system 1 or 2? Does that help? Could be 2. It's 1. Okay? And so photosynthesis, really fast. Light comes in. It excites photosystem 2. Electrons start flowing. The water reacts with manganese center to take electrons out, gives it to the tyrosine molecule. The tyrosine molecule gives it to the P680 in photosystem 2. So I go through all the channels in photosystem 2. I give it to Q. Q goes to cytochrome BF complex. Protons are pumped across, and electrons are given to plastocyanin. Plastocyanin goes to photosystem 1. Light shines. What wavelength excites photosystem 1? Yeah, P700. Electrons flow through that. It goes to the ferredoxin, and the ferredoxin is supposed to go to the NADPH reductase to give the electron so that NADP plus can pick up the H, the hydride, and create the NADPH reductive. But what happens when I run out of NADP plus? Where do the ferredoxin electrons go? It can go back to the cytochrome PF complex. That's one pathway, and that can make a little bit more proton gradient to make more ATP. Also, the ferredoxin goes and it upregulates several enzymes in the regeneration pathway for carbon fixation. Okay, and so it has to reduce those enzymes to make them functional. Now we know that ferredoxin can go all the way down to the roots, translate, and give the electrons to break down nitrogen. So ultimately, the electrons are coming from photosynthesis or the sun. So this is where all of those electrons come from. The nitrogenase actually has a cluster that holds on to the electrons for just a little bit to be able to transfer over to the iron molybdenum cofactor, and which the nitrogen actually is having the reactions with the electrons and the hydrogens at that particular place. So we have a P cluster and an M cluster. The M cluster actually is kind of interesting, okay? If you look at this side, this comes up towards us. This is a sulfur here, and this is the iron in the iron. So from this side, the iron is up. From the back side, it's kind of open. So this is that same cluster flipped over. And this iron, that iron, that iron, this iron, that iron, and that iron make, well, actually, this iron and this iron, that one and that one, are all involved in coordinating the nitrogen that sits. That sulfur that is now down below is the one that's sitting up here in that structure. So the nitrogen fits down wonderfully. How many electrons does nitrogen have to absorb to get to the ammonia? Go back a few slides. Not the overall process, not the one with the side reaction, but directly. 6. 1, 2, 3, 4, 5, 6 irons. Every one of those irons is going to donate an electron to the nitrogens. As they donate in a stepwise fashion, it will pick up protons on the way. And so electrons will be donated, protons will be picked up, and you will eventually break that particular bond. And it comes off as ammonia, and as soon as it gets into the solution, because of the pKa of ammonia and the pH of the buffering system, it will become the ammonium ion. What did we learn in the degradation of protein about ammonium? Is that good for us? It's poisonous. Okay? Even at 60 micromolar, it becomes poisonous. And so we did learn one particular example of an enzyme. In degradation, it was focused this direction, and that we will create an intermediate and we will have the nitrogen fall off. But in the synthesis, we will do the reversed reaction and we will create glutamate. All right. this right here was a particular structure that we needed to know the name of. Do you remember what the name of that particular structure is? It's a functional group. We called it, it's not exactly, we called it something different in biochem because it's used quite often. It is reversible. We can add water across that. Schiff's base. And so the Schiff's base is used a lot within biological systems, and you should be familiar with that particular intermediate. Okay, interestingly enough, we said that reductions, and that's what's going to need to happen in the synthesis here. We have to reduce that. We're going to use NADPH almost all the time, although this particular enzyme can survive by using NADH. And we usually use NAD plus as the accepting. All right. Interestingly enough, when you attack this, NADPH, and if you use this, you're going to attack at the carbonyl. Even though there is a positive charge on the nitrogen, you will actually have the hydride attack because the intermediate that is temporary is a neutral nitrogen. And then because of the pKa, you can pick up the proton. I would write it as a movement there. So this is 1, 2. You could write that. That's perfectly fine. Or you can write that movement. That is saying the same thing. Now, the dehydrogenase is the reason why we only synthesize the L version of the amino acids. And so if you go back to the Miller experiment where they synthesized amino acids using the what was determined to be the primordial atmosphere and used electricity to simulate the lightning strikes, the synthesis of amino acids actually happened, but they actually made them in a racemic mixture. And what we find in the human body is only the L versions of the amino acids. The reason why we have the L version is because all of the amino acids are synthesized with the dehydrogenase. And so on one face of the substrate, the amino acid binds, and here is the protein. The protein aligns the hydride that's going to attack, okay, and it only attacks one face. And so when we have the ability to attack something that is flat, okay, we can attack either side in an organic flask, but because the enzyme forces the binding and the positioning between the two entities, we will only get one stereogenic outcome, and all of the amino acids are L. All right, amino acid synthesis overview. Oh my goodness gracious. There is a lot of synthesis going on here. Okay, what you need to know from this is which amino acid fits into the starting point of a pathway. And so this group right here comes from oxaloacetate. Okay. This group over here comes from alpha-ketoglutarate. Provate will make these 4. 3-phosphoglycerate, make those 3. The erythrose 4-phosphate can help make tryptophan. Phosphoenol provate will also be involved in phenylalanine, tyrosine, and tryptophan. And glucose 6-phosphate will go for histidine. All right. Name what pathway, oh, sorry. What pathway is this molecule in? Glucose 6-phosphate. What pathway are we in? Glycolysis, half right. What's the other half? Not citric acid cycle? Gluconeogenesis. Okay, so we can go either direction. 3-phosphoglycerate, what pathway? Glycolysis or gluconeogenesis. Urethros 4-phosphate, what pathway? Pentose phosphate pathway. Pyruvate, okay, that one's glycolysis, right? Gluconeogenesis also. Then oxaloacetate from the citric acid cycle, alpha-ketoglutarate from the citric acid cycle. If you went through Biochem 2, you learned these associations and the degradation. The synthesis is no different where they come in and out. Okay? And so hopefully this is not relearning that entire scheme. All right? And so let's start and do one of the most simple synthesis in the pathway. Now, you have learned how to synthesize one amino acid at this moment in time. Which one? We have seen the synthesis of one amino acid. Did it go by you? Went right by you. We know how to synthesize glutamate. Alpha-ketoglutarate plus the ammonium. Okay, so you've got 1 of 19. Here's number 2. All right, we're going to do glutamine. What we're going to do is we're going to add an amine to the R chain. All right, when you look at an enzyme reaction, okay, and so this could conceivably be a exam question. All right, we're going to synthesize this glutamate and this intermediate. Okay, I'm going to give you one step in a process. All right, so this reacts with ATP. ADP is the leaving group, and I have this phosphorylated intermediate at the end. What in the world do you start doing? What's a strategy by which you approach these? Okay. Base. A lot of the times a base is involved, and you also have an acid, because you always have an acid and a base in a biological system in the enzyme. So you might start writing a base down and throwing electrons around, throwing arrows. I would do one thing before I start just drawing arrows. Okay? First thing I like to do is do some inventory between the beginning and the end to find out what changed. So I look over here. If I look at the reactant and I look at the product, any changes around here? Nope. Changes around here? Nope. It changes around here? Nope. Changes around here? Yes. Okay. Did I change anything here and anything there? Let's see. What in the world's different? Okay, so this oxygen is bound to that carbon, right? This oxygen is still bound to that carbon, but what do I see? Double bond. Where do you see the double bond here? It's doing what? It's in resonance. So to facilitate the end, I'm going to stop the resonance. I'm going to say, okay, if that is a double bond, then I must have a negative charge here. Okay? And then what we see is this oxygen is right there, and that's got something different happening to it, right? It's got a phosphate attached to it. All right. So I know something's going to happen here. All right. ATP. ADP. Does that tell me anything? What does it tell me? It's going to attack a gamma phosphate. Okay? You can tell where the ATP is attacked by what is the leaving group. Is it ADP? Is it AMP? Is it PP? Or is it inorganic phosphate, the 2 PIs, and the diphosphate coming off? And so because I see one phosphate, I know I'm going to attack the gamma. And I don't need a base. Why? I already have a nucleophile, right? a negatively charged intermediate. We'll attack at the gamma. Every time I attack the phosphate, what do I have to do? Because I have a pi bond, the intermediate always has to be a full charge on one of the atoms temporarily. That's the high energy state that has to be stabilized. That's the excited state. Things don't like to stay in the excited state, so I'm going to kick down. And I'm going to show the bond breaking. I take this pair of electrons and I put it onto the oxygen. That's the ADP leaving. And I have the phosphate at the end. Putting that phosphate onto that intermediate, why in the world did I do that? What does that afford us? What does that give us? A nice leaving group. The phosphate's a wonderful leaving group. One other way you can describe it is I've activated that molecule. And what I've really activated is this. If something attacks at the carbonyl next to it, then that phosphate can be a wonderful leaving group. Now what will attack it, if I take a look at my inventory, is here, carbon is bound to oxygen. Here, this carbonyl is bound to a nitrogen. So I've got to get my ammonia. Okay. Also, here 2 hydrogens, here 3 hydrogens. Okay. So what I'm going to do is I am going to use a base in this case. And ammonia, the lone pair is a very weak nucleophile. Okay. It can attack, but if the base takes off that proton, I now have full negative charge, which is a strong electrophile. Okay. Sorry, a strong nucleophile that it can attack the electrophile. I kick up, I kick down, that pair of electrons goes off. Phosphate is the leaving group, and now you've made 2 of 20 amino acids. Okay, in this slide, this projector, you can't really see, but what do you see in your slides here? Little what? Arrows, dotted lines. And all of this doesn't mean X-Men, okay? What does this mean? Negative inhibition. So to go from glutamate to glutamine, right? All right, what turns it off? CTP, AMP, histidine, tryptophan, glucosamine 6-phosphate. Oh, carbamylyl phosphate. What in the world is carbamylyl phosphate? What pathway? The urea cycle. Okay. Ooh, alanine and glycine. Okay. All of these are downregulating. This is a pretty important enzyme because there's a lot of allosteric effectors. Okay, what if you have one signal that turns it down? Is it going to turn it off? No. This enzyme almost never gets turned off. It is always sending something to some pathway. Now, if you need every one of these things, it's going to be working really, really fast. If you only need one of these things, it might only work a portion of it. When we first tell you about enzymes, we talk about being on and off, being in the on state or the off state. Well, like you think about it as 100% on or 0% off activity. This is an enzyme that will be able to be varied all the way through from 0 to 100%. And it's always working at some level of intensity. It just depends upon what pathways are being used and what kind of information that we need or what kind of starting material that we need. Okay. If I take a look at the amino acids in synthesis, we talk about the essential and non-essential amino acids in humans. Now, depending upon the book that you use, there are either 10 or 11 essential amino acids. This book tends to do or give you 10 are essential. It is this one right here, tyrosine. Okay? We do not have the enzymes necessary to make tyrosine from scratch. But what we can do, if you have phenylalanine, the first step in its degradation is to make tyrosine. And so if by chance you have phenylalanine as a starting material, not the smaller molecules, we do have the enzyme to make tyrosine. So this is the one that's debated about whether it's essential or non. This book is going to tell you it is not essential. Okay, so we can make 10 of them. All right. And we have seen the synthesis of this one and that one up to this point in time. All right. The others we have to ingest. All right. And it's kind of interesting, even in tyrosine, phenylalanine is also a essential. So we have to eat that one, and then from that we can get this one. All righty. Who went last? Who did you pick? Oh, Noah. Yeah, who did you pick? Bree, come on up. All right, so we have synthesized 2. We're now 3 of 20, and we're going to make proline. Now, we're going to start with glutamate. So we know where glutamate is. All right? Okay. Now, I know when you come up here, you start feeling panicky, right? But here you go. What you're supposed to do is what? What's the first thing? Other than cry.
Okay, that's on my side.
Yeah, you got to look and do inventory. So that's new, right?
Yep.
And that comes off, so that gives you an idea. Do I have to deprotonate anything?
No.
No. Because there's something already negative that can be the nucleophile, and where will it attack?
Almost phosphate.
Okay. Oh, pick a color, any color.
Green.
Green. Up, down, and out. You are done, or you're halfway done. Now you got to pick somebody. This is the social pressure of the job.
Jada.
All right. Why did we do this step here? Why did we do this step here? What do we, come on, Jada, you can come up. Yeah, right now. Why did we do this step? What do we call this? We've activated it, right? We've made a good leaving group. Okay? Now, I'm going to do some inventory. One side there's a phosphate, on the other side there's not a phosphate, right? It's got to go off. Okay, what's added in its place? Not just a hydrogen, a hydride. What else do we have that is there as a reactant? NADPH. So, from the NADPH, we get what organically? A hydride. All right? And hydride will be negative, nucleophile, and it's going to attack something that is an electrophile. Okay. And what happens when you attack a carbonyl? Up, down, and out. Very good. Now you get to select someone else. Okay. So, it was once a acid. Now what is it? What's its functional group? Aldehyde. Okay. All right. Who'd you choose? She's calling you out. Remember that. All right. Now, the glutamic gamma semialdehyde doesn't look like proline, but we're going to get close. Now it all gets crazy.
Yeah.
Right? Does it look crazy?
So would I deprotonate
Yes.
and then come attack
Yes.
and then grab an H?
Mm-hmm.
Perfect.
Now, yep. Okay. Ooh, nope. Reverse.
There it is.
Don't reverse too much or you're going to have to...
What I'm going to do is,
Yes, that's what I would have done too, because it's lazy.
yeah. There it is.
Good. All right.
Yeah.
And so we have closed the ring. That's starting to look a little bit more like proline, right? Who are you going to choose next? Ah. She's going to remember that. All right. And we're going to get real close now. Very good. All right. Choose someone else for the class at this moment in time. She just wrote that mechanism. Is it a substitution or an elimination? Is it an E1 or is it an E2? It's an E2. Very good. Who did you choose? All right. All right. Now, what do we see? What is a reactant that we know? NADPH, and so we can use a hydride. And now, what's not going to happen on the exam, it's not going to be color-coded. One, I'm not giving it away, and two, it costs twice as much to photocopy something like that. Okay? So, you know a hydride is going to come off the NADPH, and where is it going to attack? Yes. You can put your hand on it. Yes. Well, it can go to the nitrogen, but look at what happens to the nitrogen. Mm-hmm. Right. And every time you do an organic mechanism inside an enzyme, you have a base and you have a proton. But remember, they're not just a base and proton roaming around. They're one of the amino acids that are on the inside of that enzyme. And you have synthesized proline. Wow. Okay, choose someone else. Okay, so we've synthesized glutamate, glutamine, and proline. 3 of 20. All right. Next. All right. We're going to start with 3-phosphoglycerate. What, I didn't ask this before, but what pathway are we in with this starting material? 3-phosphoglycerate. Glycolysis. Very good. So we're going to start and take molecules from glycolysis, and we're going to make 3-phosphohydroxypyruvate. Well, what is different between the glycerate and the pyruvate there? It's all around that what was once a hydroxide and is now a ketone. What else do I have organically as a reactant? NAD+. NAD+ is a what? An acceptor of a hydride. So hydride can leave as part of this. Yes, that's one way to do it. Let me try, go pick someone else. Okay, that's another way to do it. Okay. Here, this first movement, number 1, will place a negative charge on a carbon. This movement here, number 1, will place a negative charge on a oxygen. Okay. The enzyme could make it go either way. Okay, we would have to do some testing. But organically, which movement would be less energy? One with the red or one with the blue? One with the red. Okay? Now, not saying the enzyme couldn't do it the other way. The enzyme would have to extraordinarily stabilize that anion, or it could do it the other way. And it still has to stabilize that oxygen. All right, who's the volunteer? Alex? Oh, you know what? You got saved. All right, Alex, you remember. Okay, you have this slide right here when we come back together next time. You get a little time to think about it. All right? Well, just wait as we go through. All right? So, what I want you to do is to look at the quiz for this chapter uploaded. It's going to ask you to do multiple choice questions. It is really an integrative process about what's happening. I want you to answer the question. Give me a letter. Give me why, which is the most points, and actually give me a reference for each one of the questions that is answered. So it isn't just guessing. Guessing is going to be a very small part of the circle the letter kind of thing, right? The actual reason why it's right is important, and then the reference. Where do I not want you to reference? Wikipedia. Even though it is better than encyclopedias, but it's still, I want you to be a little bit more in depth, okay? And so start looking at that quiz. And look at, if you were unsure about the organic mechanism, watch the video.